Hey guys, welcome back to Bioinformatics. We're going to finish up our microarray. Well, we've actually really done the microarray experiment. We have our log full changes and which ones are significant. But it's important for a computational biologist to put it into biology, right? So if you're a computational biologist, we've done the computation part, but now we need to do the biologist part. So we're going to use a program. There's multiple ways you can do this. Gene ontology is one way to kind of give you an idea, like the cellular components or the type of metabolic pathway. It'll just give you like a list of what they are. But I really like PathView. And what PathView does is it allows us to kind of visualize and map the differentially expressed genes back onto pathways to see if significant pathways are enriched for genes that have been affected by spaceflight. So with that, let's get into it. Okay, so we already called this PathView, and that's a good name. So we have to load a couple more libraries for PathView. So we're going to load one called PathView. We're going to load one called Gauge, which is generically applicable gene expression, I believe. And then we're going to do one called Gauge Data, which is just some associated data. And then we're going to take from that Gauge Data GeneSets.hs. Okay. So let's quick run these and load them. All right, so now let's do fold changes equals. Maybe we already did this. Oh yeah, so we already have fold changes, so we don't have to run this code again. I have it on here twice. Okay, so let's do names of fold changes equals all to entries. Oh, maybe that's why I ran full changes again. Let's try it again. Full changes equals all to log full change. There we go. Okay. I must have changed the row name at some point. All right. So let's look at the head of full changes. All right. So these say NA because some of them they should say. So if we look at where's full changes? Oh, okay. So for the ones that have entrust IDs, it'll say entrust ID. If it doesn't have an entrust ID, it'll say NA. So that wasn't very useful, huh? All right. So Keg, ATH. So Keg is the pathways that we're going to map these functions to, or these genes to. So we're going to say Keg ATH equals keg.gset, and we want Arabidopsis, and the ID type equals entras. So what this is doing is it's using some of this Keg data, and it's saying map using Arabidopsis as our organism, so it knows what database to access and to look at, and that when we map, we're using entras IDs to map it to the pathways. Let's do Keg ATH Sigmet, Keg ATH KG sets, Keg ATH Sigmet IDX. So I run both these. Keg ATH not found. Why didn't it do that? Could not find Keg GSet. GSets plural. Okay. All right, so we should have Keg ATH here. So as you can see, it's just kind of a database where it says what our gene is, kind of an annotation of it, etc. And then when we run keg.ath, is this the right one? Sigmet? We've just kind of pulled it out, right, from that database. And let's get the results. So Kegrez gauge, it doesn't say, give a description here, but it's that gene expression function. Full changes is what we're going to use, and the G sets equals, the gene sets that we're looking at are the Keg.ath Sigmet genes, and same directory equals true, which means that we've saved everything in the same directory that we're working in. So we don't have to give it a different place to search for. Okay, I'll apply Kegrez head. So we're looking at, this is just looking at Kegrez, the head of all the columns. So you can see here we have the pathways. So the ribosome pathway is, the Q value is our multiple test corrected value. So this is super significantly change in expression, up or down regulated. So this is greater. So we've pulled out the upregulated and the downregulated in this analysis. So this means that in space, ribosome, in photosynthesis, and all these things are increased in expression. And the pathways down here, none of them are significantly down-regulated after we do multiple test correction. Some of them are, it's still good to look at, because you also don't want your stuff to be too stringent and you ignore something that might be a biological phenomenon just because your stats put it out. It's always good to acknowledge it, but treat it with skepticism, if that makes sense. Like, you don't want to say, oh, this is fine in space. Well, it might be downregulated a bit because our p-value non-corrected was significant, and we have to acknowledge the shortcomings of our experiment. We only had 3 replicates for each, right? 3 flight and 3 ground control. So this might be because there's enough error in the biology and the experimental design that we couldn't detect this. Like, it was close but not fully detectable. So it's always something to look at still. You should always not just completely ignore 100% the stuff that's not significant, but treat it with a healthy amount of skepticism. You don't want to be like p-hacking where you're like, oh, I'm going to change my analysis so this is significant now. But it's always something to keep in mind, like how you digest your data and what you consider interesting. That's kind of up to you as a scientist. So we're going to create a variable called grader, and this is the Kegrez grader. And then we're going to create lessers, which is Kegrez less. All right, so this is what screwed me up for a while. Well, one of the things that screwed me up. Like I said, I tried to make these videos multiple times. I don't know why they said greater and then less instead of lesser, but it's less. So make sure you put that. And so now we're going to write these to a file. We'll call this brick 16 PathView grader pathways.csv, and then we're going to do the same thing. I'm just going to take copy and not paste. I'm going to copy and then paste, and I'm going to do lessers, and then call this one lesser. Okay, so let's run this code. Lesser's not found. Kegrez not found. Oh, it's Kegrez. Where did I get Kegrez from? Kegrez. Lesser's not found. Keger's not found. Kegrez. What am I doing? Kegrez. There we go. Jeez. There. Okay. So those files should be written to a CSV. So if we were to go into our scripts here, I should have brick 16 PathView Creator. So this says all the pathways. As you see, there's a lot of pathways that were. This is great array. So upregulated, and then it gives us our statistical mean, our p-value, our q-value, which is what we really are interested in. Set size is the number of genes in these pathways. So sweet. So we now have a list of everything, but that's not the end-all, be-all. So we're going to take this a little bit further. So now let's, what should we call this? Let's map two pathways. All right, so Kegrez, two G's in Keg. Kegrez pathways equals data frame. ID equals row names. Kegrez Creator. Kegrez Creator. And then let's pipe it down. So we'll actually use a tidyverse function here for once. So we're going to do a table data frame. Pipe it down. Character. Okay. Let's run this real quick. Okay. So, what this is doing is we are creating a data frame where we take the Kegrez Creator data frame, or data from Kegrez Creator. So we're taking just the upregulated stuff and we're turning it into a data frame, and we're filtering out we're just taking the first 5 pathways, right? And then making them characters. And then, so now if you look at Kegrez, Kegrez pathway, right down here, you see it's just the names of those pathways. And the reason we do this is because we're going to be generating images, and we don't want to generate like 1 million PathView files, or like every PathView file. Ideally, you could filter it just based on which ones are significant, but we'll just pick the top 5 for this. So, Keg Resids Creator. We're going to make a substring of the Keg res pathways. Start equals 1, stop equals 8. So what we're doing here is we're taking our Kegrez pathways, and as you notice it says ATH whatever, then photosynthesis. Well, this command, the substring, it just takes out the names, because all we want is, they look like they're gene names that we saw from the Terra database. These are actually pathway identifiers. So ATH03010 is the annotation for the ribosome pathway. So for mapping this, we just need to give those. We don't need to include the actual characterization of what the pathway is. Okay, so let's do gene data as character all to log of C. We've already saved full changes, but we've messed with it, so we're just making another one. So let's define a plotting function to apply next. So we're going to make a function here, and it's going to be called plot pathway, and the function, we'll call it PID, is PathView gene data equals full changes. So we're using the PathView function. This is to map onto these pathways, which gives us really cool figures. And we're using full changes to tell which genes are differentially expressed. Pathway ID equals whatever we call our function on. Species equals Arabidopsis, so make sure it maps to the right pathways. And then new signature equals false. There should not be a comma after PID here. Okay, so let's run that. Okay, so we have our function now, and so let's do, so we can use that if we want to go back and plot other pathways. So plot multiple pathways. Plots are saved to disk and returns a throwaway object list. So it's going to make a temp file while it's writing it. So we'll say temp is sapply, so apply to all of these 5 pathways. So it says go over the list, Keg Resids Creator. So go over everything in those 5 variables and use them in the function PID, and for this function we want our variable set as PathView equals gene data full changes pathway ID equals PID, species equals ATH. There should be 2 of those. Okay, so let's run this. Error. What did I do wrong here? Gene data, full changes. PID, species equals ATH. Function. There we go. Typos. There we go. So see, now it's going and it's getting gene IDs, using those ATH numbers from up here to look in the Keg data set, and it's taking these image files, and then it's mapping onto these image files our gene expression data. So what you'll get is, we have to actually open it up in your file right now. So if we look at this, we have the photosynthesis pathway, and this is all the genes in the photosynthesis pathway, and the red ones are the genes that are expressed more in space. And so you can see we have this nice schematic that kind of shows us what the photosynthesis machinery looks like in a plant, where we have our This is antenna proteins, but we have our ATP synthase, excuse me, photosystem 1, photosystem 2, and the antenna proteins would be up here. There's a separate figure for antenna proteins, I believe. But it shows you, so photosystem B, subunit Q, subunit R, all these things are turned up or expressed more in plants grown in space. And so we have this for all these different pathways. So this is linoleic acid metabolism. You can see a lot of things are turned up, but some genes are turned down. The green there turned down. We have carotenoid biosynthesis. So you can see a lot of them are turned down. And the intensity of the red is how high or how low the expression is changed, right? Oh, I guess, okay, so this is an interesting figure. So it must be the green. Oh, I don't know why that one's, Keg's kind of weird on that one. So you can see for ribosomes, almost everything is turned up. And if you watched the lecture from Dr. Costas, Sylvan Costas from NASA, they found in mice and stuff that ribosomes are greatly affected in spaceflight. So this is it might be something ubiquitous to all ribosomes. It's interesting. So yeah, okay. So you can take some time to look through those if you want. Now let's do one thing here quick. Let's take this data for all these scripts. So starting at, let's change this annotation. Let's map to pathways greater, and copy all this and just paste it again down below. But let's change it to lesser. So now, wherever there's a greater, we call it lesser. Lessers. 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 Okay, so now if we run the exact same code, but instead of pulling from the graders list, we're going to look at the pathways that are turned down in spaceflight. So just by changing those things to lessers, we should be good to go. And Kegrez lessers. Did I not? Oh, nope. Sorry. Wait. Oh, we need to call it less. Just less. Less. Just for those, because in Kegrez it's called less and not lessers. So if we were to look at Kegrez, see it says less, not lessers. And it's going to pull these out. Okay. Oops, let's make sure we select this K. Let's try that one more time. Kegrez lessers not found. Where else does it say Kegrez lessers? Oh, we need to change this to less first of all. Kegrez pathways. Kegrez less. Kegrez less. Kegrez pathways. Kegrez pathways. So what I could do is there's a little magnifying glass here, and in sections, so make sure you have it highlighted. I'll just look for great. Okay, it says there's nothing there. X and selecting method of function as apply object to Kegrez. Keg Resids lessers not found. Where did I call Keg Resids? Keg Resids less. Okay, let's try this. Let's try running it again real quick. Keg Resids lessers not found. Oh, right down at the bottom here is where I'm missing. Okay. All right, there we go, finally. Okay. Okay, so we should have more pathways here. All right, here's an example of, let's say, MAPK signaling pathway for the plant. You see a whole bunch of green on this one. So these are ones that are turned down in spaceflight. So you can get an idea. Protein processing in the endoplasmic reticulum, lots of green. This one's kind of in between, right? Up and down, which is kind of interesting. But that's it. So from here, we're not going to get into the theoreticals of what this means for spaceflight, because that's a big debate and that's my area of research. So yeah, but we've taken those numbers in the data set and we've gotten an idea of, okay, what are the pathways that are affected? So what, not just a single gene, but what biological processes are affected? Because in the grand scheme of things, genes are important, but genes working in a group as a pathway is what really affects how an organism survives or thrives. A single gene can affect the pathway, but knowing how the entire pathway responds to a unique environment, that's really important, and especially when NASA's goal is to optimize conditions for things to be able to survive in space. Knowing that photosynthesis is affected, maybe use different lights or different oxygen, carbon dioxide buffering, or things like that to optimize it from a physiological standpoint. So that's all we got here for microarrays. We made it through. I didn't mess up a single time besides my typos and searching, but I didn't have to restart. This was money, Isaac. So all that being said, I hope you enjoyed this. Next, we're going to get into newer technology, RNA-seq. So microarrays are, like I said, not used so much anymore. RNA-seq seems to be the present. There's probably going to be something even greater in the future. But let's process some RNA-seq data and see how we do with that. So thanks for participating.